I'll wait for Giacomo to be back. Let me know, Giacomo, when you're back so that I can ask you a question already. Oh, you're back.
Yeah.
I just want to break the ice by asking you one thing on, you showed the table there, slide with lots of different ways to share the data, different standards, and I understand that, there are different technologies, there are different areas of the field that are looking into different stuff. But like, imagine that now I come up with a new standard or whatever that is somewhat not acknowledged by the field quite yet, by the journals quite yet. Like, it sounds like the journals have control over that, the big journals. Is it common that there is a new standard coming in there, or is the field kind of stuck to those main ones?
So I would say that it depends on the kind of data that you are trying to standardize. I say it better. So at least in my field, the enabling technology was what is called the sequencing technology. Roughly speaking, the fact that you put a biological sample as input, you do some preparation, and as output, you have some text file that describe the genomic material of the input sample. So let's say that the output since 2009 is strings. Okay, so if you put in input DNA, you have DNA string as output. If you put RNA, you have RNA string as output. I mean, I'm oversimplifying, but just let's say that the key point is that the output data of the machine, of the measurement tool, is more or less the same in terms of the format, strings. What it changed in our field, for example, is the length, is the error profile, etc. So the format that I show you, the first example, what I call the ARD file format, is something that is in the analysis pipeline is quite close to the raw data that comes out from the machine. So roughly speaking, is the format that allows you to tell you, okay, that string that is the output of the tool, from which portion of the genome come from. I mean, again, oversimplifying, hopefully there is no bioinformatician here in the chat, otherwise I will be killed by the people in my field, but let's say that the oversimplification could be this. And so for this reason, is quite established because is trying to put a formalization of a quite obvious concept. It's just, they pay often attention in the quite complete, even having some field in the file format that could be, useful for the future. So this is something that should be always be considered when you define a standard. Think not at the data as now, but it could be in 5 or 10 years. So look at the future. And but this kind of format is just is a matter of being more or less complete or more or less efficient. But there is this SAM or BAM that I show you. There is probably another one that is more, let's say, an alternative version, that a true alternative of the format. So for raw data, there is just very, very few formats. The variety of format are what are called the soft format, soft data format. So the one that try to standardize data that are more far away in the pipeline. In bioinformatics, we talk about analysis pipeline, because starting from the strings that come out from the measurement tool, you have at least, 7 or 8 methodological steps, and each of them, one is denoising, one is annotating the data. So there is really a lot of steps. And for each step, basically, there is some data forms that are more or less used. So the one that I call softer are the one more related to the downstream part. So where really you are more focused on trying to answer your question. And so even the data format is more a way to helping you to have a very compact, clear way to store the data, but is even if you want more oriented to the kind of question that you want to answer on that part of it. While the first one, the one upstream, is just a way to store strings in an efficient way. So you're just character. You are not able to tell nothing about the biology. You are just a way to store string in an efficient way. So I think that this is the true difference between our format. Very few of them, very established, and it's very difficult if you provide the new one. You publish a new one, it becomes the leading standard because you should provide a lot of improvement. While it's more easy to the downstream part of analysis.
Yeah, I suppose in my head, and then there is a question there. with the hands up. In my mind, I'm trying to be very concrete and thinking about the parallel between what you are talking about and our situation, where we have different EEG devices and then different MEG machines, and so they really have different formats for specifically for the device, and then you jump to a more specific, to a standardization of some kind. And as Liberty said in the first day, it's still the Wild West, and that's where we are trying to give a direction. And go with the question there, Bjorn.
Okay, yeah, so I just wanted to elaborate again on the question that I asked already on the chat. So thanks for asking the first part of my question. So it sounds like legally it's possible, let's say, to publish such data. But I was also referring to, let's say, about more the ethical concern. So because I'm wondering if Amazon is hosting the server, I mean, Amazon is not a charity organization, right? They might have some interest, who knows what and whether they actually do have. But I'm just imagining a scenario now where some, let's say, insurance company would check the gene database, and depending on your genes, whether you have any prevalences for any diseases, your fee would be adapted, or they won't give you an insurance because of your DNA. It's just exaggerating. I'm well aware of this, but just wanted to know how the field of bioinformatics generally deals with this.
So I will start from the last part. So if an insurance company is trying to querying somehow the data and identify which, I mean, from my knowledge of biology, there is nothing, like such kind of biomarker. But let's say that it exists, just to make, the example complete. And identify that I'm more, I have a higher probability to have that particular disease, and making me the fee of my insurance higher for that particular stuff. Well, this is possible if that data in that repository, it could be associated to me. I mean, Giacomo Baruzzo, not just a guy coming from Europe. So, and it's still the part related to, anonymization. So if the data are there and are even very complete, but there is no way to tell that that sequence is my sequence, the insurance company, we know that there will be a guy in the 8 million or 8 billion of people in the world that maybe have that high probability to have that disease. So, I mean, in this sense, is the concept of anonymization. I mean, it's not bad that the data is there. It's bad if that is there and there is the way to associate that data to a particular person. Otherwise, you have just, one sample from distribution. Is nothing, is nothing that it could be identifiable in any way. So, I mean, as I told you before, I have to be honest, I'm not both a biologist and a lawyer, so I'm not able to tell you, the bound, so the amount and the precise kind of information that make that particular biological feature, very informative and univoc for each person. So I would not, I'm not making an example because I don't want to say something that is not accurate. But for sure, if the anonymization is doing right, is possible. So when I was in the US, I did a test which is called, if I pronounce it right, HIPAA, so it's HAPPA, something like that. And this is a way to test if people that work with data really know what it means that anonymization make a sample not, linkable somehow to a particular person. And was an example to say, okay, if I just have the information about that particular patient, and I put just, the city where these people live. Well, if it's a rare disease, and in that city there is a register of the people, even if I don't put his name, I could have a way that is not varying the data, but is do some mining of the data to connect that sample to that guy. So, I mean, I'm not saying that it's easy to find the boundary of both, which is the amount of biological information that is identifiable, and the amount and type of covariates that with some processing make that amount of information identifiable. So, I mean, we should talk both with a biologist and a lawyer, and even, with a good data miner probably in the middle. So, but for sure there is the possibility to do, because otherwise we have, half a million of example of out of law data in the representation. So there is this way, this boundary. I mean, I'm not a biologist, not a lawyer, so I'm not able to do precise sample, but for sure there is. Otherwise, you know, we are all outlier of the people that put data there.
Bjorn, does your question go beyond like privacy and identifying like brain data or genomics data? Like, do we give companies that are out there to make money like our data that they can use for that? Is that part of your question, or was it all about privacy and identifiable data?
I think it's both. So also the concern that, I mean, we had this discussion in our lab as well, that if we give the decision to our participants saying, you can agree whether your data will be shared on an open repository, they might not be aware of what can in the future be done with this data. So one point, the identifiability, I mean, there's always good and bad things that can be done with it. I mean, having lots of data in many cases makes things easier and gives us progress. But no one really knows what can be done with the data as well. So that's also part of my concern. Yeah.
Just the people aren't completely informed. Maybe they can be completely informed.
Yeah, I mean, even for us, we cannot say, like we being the, let's say, EEG experts, for example, even we cannot say what can be done with the data in 10 years. So how should a participant be able to do that?
Yes, that is a good point. So I had a question actually. As far as I know, I don't really know much about genomics data, but it seems like the data that you acquire in genomics or any of the omics is pretty closed-ended, in that you are looking for the presence of, for example, like base pairs, and there's only 4 or 5 of those, right? And so there's not really too much measurement error. There's not really too much bias in my head, at least. But in neuroscience, we have a lot of those sorts of things. So we have issues like noise. We have issues like experimenter can bias the participant. The experimenter can bias the analysis. Things like that. And things are much more open-ended. And I was wondering if there's anything like that with the data that you, so the problem I see with this is that if you have these open-ended data formats, like how do you know which data to trust? Like you share a bunch of data, which data are good to use? Which do you leave it to sort? Do you leave the community to sort it out? Is there any issues like that you have in genomics data?
So when we talk about the raw data that comes out from the machine, so the strings, the machine assigned to each single character in the string somehow probability it is right or wrong. So it's up to you to be more strict in the filtering, so remove a lot of data or so, but it's something that comes with the instrument too. So with the measurement tool, we provide you the string and the quality of each single character. So, I mean, you can apply more or less strict filter, but then it's up to you. Typically, when you upload the data, it's required that you upload the raw data, such that each one that would to download and reanalyze the data can use his own criteria of filtering. So be more strict or be more relaxed, whatever. So typically the data that, for example, are uploaded as raw are typical raw data. You just upload the data, and the quality of each single character. Then it's up to me that download the data, or you apply different filters. So in this sense, you upload the original data, and then it's up to the user to decide what to do. So in this sense, is demanded to the particular reanalysis of the data define the quality and the threshold for the quality of the data.
So it does seem like there's a little bit of measurement, maybe not error, but like ambiguity. Is there anything where like an experimenter can bias the results that you have to deal with? It seems like those sorts of things are pretty would be pretty common or could be pretty common in neuroscience data. I'm just wondering if there's any sorts of tools out there or like procedures that we might borrow.
So if you're talking about something like batch effect or contaminants or whatever, sure. So the first study, especially with the first technology, we see that there is a quite good association between the technician that was collecting the DNA and the sample. So, I mean, something like that. Then, you know, protocol becomes more stable, and, you know, the room temperature of the guy that was looking at the sample, you know, becomes less and less evident to identify as batch effect. But yes, there is this kind of issue, as well as, you know, obviously the machine are doing, there is some error in the data that is produced. So there is still a lot of issue. So for example, there is one of the first step of the preprocessing is even in our field called denoising or missing data imputation. So there is something like that. The problem is that we, at the beginning, work with strings. When we moved with integer, because we just, at least in some of the field of bioinformatics, count how many strings come from a gene. So we even change the type of data. We start from character, and with that, with huge table of number. So, you know, but at each step, there is this kind of error that come from the instrument, that come from the guy that did the experiment, batch effect, environmental covariates. So really, the temperature was something, for example, that, you know, could have a huge effect when you are collecting some samples, so the temperature during the processing of the sample. So really, there is a lot of possible bias and other side effects, negative side effects.
So it sounds like we just leave it to the community to sort out, just give everything or share everything and people sort it out.
Yeah, I mean, there is obviously a huge effort in developing tool for denoising. So when you have to do denoising, you are not applying your own pipeline. There will be, I don't know, 30, 40 very established tool, because obviously, you know, the side effect of having such amount of data is that even you have a huge amount of tool to do this kind of process. Each step of the pipeline, you have at least, I would say, 20 alternatives to do it. And I mean, I'm not talking about tool published, you know, in predatory journal, in journal, something like that. I mean, tool published in the probably most prestigious journal in the field. So talking about tools that have a lot of users using them. And, you know, is the side effect. More data means even more competition. You have, you know, more way to test that your data is good. And so even, you know, more easy to publish a denoising tool, a mapping tool, a filtering tool, whatever in your data. So, you know, it's a lot difficult to publish in bioinformatics. Not only because you have, as Giovanni is saying, spend a lot of time to, you know, fulfill all the requirements, but it is a lot of competition. So
thanks, Giacomo.
I'd like to give a comment that transitions to Judith's questions in the chat there. You know, I think that by looking at what you showed in the presentation, like it's important to acknowledge that there is a change happening. And you know, I'm not old, but you know, 10 years ago when I started, I remember talking with a colleague about data sharing, and I was accused of being naive, because they were like, yeah, you know, sure, you can share the data, but how do you share it? And what are people going to do with that anyway? Because experiments typically, you know, traditionally, they're very specific. But now we're starting to have more general purpose sort of data sets with these paradigms. And one thing that Judith says in the comment there is also the importance of acknowledging the effort of data collection, which maybe at the moment in our field is not as acknowledged, and maybe one is, you know, people are not as inclined to actually spend the right amount of time on it. It's true, Judith, that there are now, you know, there is scientific data. Is that Nature scientific data where you can publish data set? There was a recent publication there on an ECOG data set. It was very little data. But there was a publication there on natural listening, ECOG, bits standardized. And so you can now also publish data sets. I suppose that you know, as Liberty said, it's sort of a Wild West. So what we're trying to do here is to give a framework that when people can publish their data, and you say, okay, I'm using this type of pipeline with this type of goal, then it gives more value to what you're doing. So I perfectly agree, Judith, and I think that's going to change, but that's, you know, it's always in the hands of the journals as well, but I think it's changing. Would anyone think differently? Do you not see the change, or do you think we should do something about it? What's your view?
I mean, I definitely know people now that are making their living just reanalyzing data sets, which is great. It's definitely changing, but I do see your point. It's like something that we talked about at COG here, actually. One of the things that we were asked to do is to give some output, and one of the outputs was how do we, or maybe chasing after a grant to collect data just for these like data hungry algorithms like deep neural networks. And so the whole point of that session was that, maybe not the whole point, but one of the points in that session was that there has to be some sort of motivation to collect data, because it's a big, arduous process, and it's oftentimes like thankless. And one thing that we came up with is just chase money to, you know, if you have money to collect data, you'll collect data. And so like we need more, maybe that might be one of the answers, to have more like grants just for data collection, just like large data sets that are standardized in some way, that have some sort of standardized like format, maybe a standardized like task. Maybe it's not even really a task, maybe it's just natural listening or something like that. Things that can be, you know, mined for, you know, findings that's not too specific to a certain experiment.
There was recently that effort, what was it called? The Manilabs EEG data collection, which was following a previous initiative, was Manilab fMRI, I think. So, and I suppose that here we're talking also more specifically about this natural speech sort of, not natural music listening experiment. So, and I haven't seen any effort like that specifically for this area, but it would be great to see, like, you know, we're doing this practically for free, but it would be great to find also a funding source that would be willing to support a Manilab sort of effort for collecting these general purpose sort of data sets. You know what I mean? Anyway, I don't want to hold up the conversation on this topic, but does that answer your question, Judith? Cool. Any other questions on this topic? There's more to come on TRF, technical size standard. What's the other title? Current challenges on TRF analysis. So if you want, we can move on, or I'll wait a few seconds if someone has more points, comments, questions, or anything. Just raise your hands. I'll give you a minute or so. Any of the in the panel, the organizers have a comment? Nick, Nate, Georgia.
I just have a little comment, and it's about what you said actually about the journals. And I have the feeling, my intuition is that researchers, we don't have the power, or we don't care, and a lot of these policies will come from the journals. You know, once the journals start making all these requirements, all the community will have to do it, right? We were talking today about, just for example, eLife. So now you cannot publish if you don't post your preprint first, you know. So it's just like if you want to publish there, you have to publish the preprint of your data. And a lot of journals now are asking for, the code of the analysis, or at least some process data sets or something. So I think, we are moving really slow. I get it about this problem of privacy is a big issue, but my feeling is also my question is until which point the privacy is our excuse also to not move forward, right? Like I think we're a community with, yeah, sharing. It's very difficult. People are jealous and people are fighting for publications, for grants, for positions. So it's no. So the goal is not to produce science or to replicate findings, to share data. The goal is to publish as much as you can. And the findings are like the second part, you know, it's not. And sometimes my feeling is also people, they don't even. So it's related with cherry picking. People delete subjects. They don't know what they did. If you ask your researchers in 3 years what they did in the last analysis, they are not able to explain to you what they did in their own code. Sometimes, you know, there's just no replication is not our thing, I would say, right? So I think sadly this will come from the journals, right? Once the journals say, okay, guys, let's do more real science, then we will have to do it. But for now, I think it's a discussion that is happening in the last 7 years from this open science movement, but I don't see so many changes, at least from the universities. I think will come from politicians, the ECR grants, or from the journals.
I even think that like a journal requiring it is still not going to be the kick in the butt that we need. I've had a couple of experiences with so-called open journals or open data journals where I've asked the authors for their data and they just flat out rejected it. So I mean, it's gonna need to be more than the journal like suggesting that you make your data available. There has to be some way to enforce that. I don't know what that would be. Like, I feel like maybe a retraction is a bit too much to have a consequence for not sharing your data when the journal asks for it. But how does the journal enforce like their data sharing policy?
You just have so many journals, and so if there is an alternative that is simpler for you to publish, you know, at the same time there is also this lie that we have on sharing data, and sometimes you just share your data in a way that is just not accessible. And so is it worth it, you know, enforcing some data sharing policies while then in fact you share something that people can't use and need to get in touch with you? And here comes the question from Jean as well. Like they say, I'm wondering what will be the common practice to establish those data standardization formats, how to make sure they would satisfy the need for most current research. And it is, I think, my opinion is that it's domain specific. And so if we start talking about a very general purpose sort of standardization, like, you know, I'm going back to BIDS, for example, which is a massive way of encapsulating a number of things. You can't really make sure that you're satisfying the needs of the research there, because it's a way to encapsulate stuff for different domains. And so that's the point of this of the CND standardization that you know, I'm trying to push for, at least within my own team, and I hope that you think it's a good idea. So if you you know, I think that for continuous perception experiments, the CND format is one answer to your question. But I also think that when you go to broader standardizations like BIDS, you can't really ensure that you're satisfying all the needs, because they're just so different in different tasks and different needs. But I don't know if you have different opinions. I think the CND is a good approach. Of course, I'm biased. But if you were at the first in the first day at my presentation, like what do you think about that? And also, Jean, does that answer your question? That's an answer in the chat. Anyone wants to speak? Don't be shy.
I have a question about the role of free and open source versus proprietary software. Giacomo addressed it in his talk. So, I mean, even if we adhere to certain data formats and then we share code, like, for example, if it's MATLAB code, right, other people might not be able to run it because their university does not provide them access to MATLAB. So recently I saw a Twitter thread where someone did like a poll asking like, well, is it legit if you review a paper and they use like MATLAB to suggest to the authors that they redo their analysis in like another open source programming language? And I mean, most people were saying no, that goes too far. You shouldn't do that as a reviewer. But I mean, I guess he has a point, right? There would be some real value if everyone can run the analyses. So I mean, what do you guys think are steps to take into that direction without like being too strict or pushing things on people which they might be uncomfortable doing?
I'll give a first answer and then let's see if someone wants to complement. I mean, I perfectly agree, and I personally want to move in that direction as well. And what we need, I think, is a way to quickly transition between one and the other without too much effort. So if now you submit something, you're on custom analysis code in MATLAB, and I ask the author, I'm the reviewer, and I ask the author, can you redo everything in Python? Well, if there is no viable alternative in Python, then it's an unfair point. While if it is a reasonable request because there are comparable solutions in Python, then maybe it's a fair request. And a couple of years ago, we did not have parallel solutions that were comparable between the two things, because maybe in Python you had boosting, and in MATLAB you had the Ridge, for example, they're particularly designed for that application. Now, as we saw in this particular workshop, now we are starting to have lots of different implementations in Python. So I think we're going to the point that it may become a fair point to make even in a review. I use MATLAB, so I don't want you to make that comment in a submission, but I think we need another couple of years to fully have a proper, simple way to transition between the two, and that would be a fair point. Otherwise, I switch journal, but I don't know what other people think.
I agree with you. I think this change will be like very natural and it's gradual, and you can switch. For me, it's like it's a change of generation. Also, the only reason I use MATLAB is because my PIs, they use MATLAB, but I see it with my generation and mostly all my colleagues and PhD friends, they are using always Python, and all summer schools or material or tutorials are always mostly in Jupyter notebooks and Python, so that everyone can have access to it. And I can see that it's changing. Even yesterday, right? We have even this Python tutorial here. So I think we need another 3 or 5 years, and then we will finally all be in open source. But maybe I'm just too optimistic and very naive,
It's good to have something that is enforced to push for this, I suppose, the alternative is always to provide a web, like another way to access the language. For example, for MATLAB, what I was trying to do there is to have a web app that can run both MATLAB and Python implementations. And so you don't need to have MATLAB because it's provided by the system itself. So in that case, it's a closed software, but anyone can access it, and the code is there. So for constrained type of applications, maybe it's fine. But as a longer term direction, I perfectly agree. Open is the way.
So if I can just do a quick comment a bit about this, we are seeing something between two open source languages that we typically use in bioinformatics, which is R and Python. So in the last year, there is a huge shift from R to Python. And the basic reason, because some tools are still not developed in Python, but still use R, is due to the ecosystems, all the libraries, all the packages, all the stuff that you have in R that you still don't have in Python. And I think that it will be quite similar to MATLAB. So as Giovanni was saying, why I should use MATLAB if I can do the same exact stuff in another language, unless I have the bias that my supervisor is a huge fan of MATLAB. But let's say if I can do the same stuff, I should move to the other alternative. The problem is that for some kind of, at least I guess it could be the MATLAB case, you don't have an alternative in Python for that particular task. And so you have two ways: wait that someone did this kind of external library, or you can do this kind of library. So very successful tool in our field are just translation of some very important package from R to Python. And this helped a lot the transition. So I mean, I'm not one to suggest what you can do, but I mean, a naive suggestion could be just, if there are very, very basic, important library in MATLAB, do the effort to write in Python, and this would be the best, push that you can provide to your field to move to another programming language. I mean, it's painful, it requires a lot of time, it could be difficult, but for sure it would be a very valuable, very even citable work that could help you do the shift.
And like, that's a great point. And like, I'm just thinking the standardization has to go beyond the language. Standardization is not about the specific language. So in this case, for example, imagine the CNSP resources and that you take them as a blueprint in MATLAB and you run all of your code for TRFs, whatever, CCA, etc. And you get your results. And then, as it was asked before, you submit to a journal and they're like, oh, but you should provide the Python implementation of that as well. You should run it with open source and license-free software and programming language. So imagine that then you can take the same files as with the EEL-Brain toolbox now that supports the CND, right? Our data structure. So you can simply take the other thing and run it again, and you don't, it's hassle-free. But that requires standardization. If there is no standardization, it's a major hassle because you literally have to rewrite the code from scratch. So that's why we need to look at integrating things, because we are not going to ask people to not write code in MATLAB anymore, because some people are just like myself are comfortable with MATLAB. So I want to keep writing code in MATLAB. But then if it is standardized and easy to transition from one and the other, it's just a matter of jumping, and if the code is standardized, you just need to rerun it on a different platform. It's a bit ideal, but idealistic maybe, but I think it will work. All right, I think we should move on to the next part, unless someone has something to say, like, oh, it's all rubbish. It's all the CND. It's a terrible idea. If you have some comment like that, please raise your hand. Otherwise, we can move on to the next thing, which was, and Giacomo, you're welcome to stay. It is a bit more specific to our methodology. And so if you want to log off, you're welcome to log off. If you want to stay, you're welcome to stay. But let me just take the opportunity and on behalf of everyone else to thank you for your presentation and your taking the time for the replies and everything. That's very useful. We may get back to you with a few questions later on, maybe on some links. If you could share a couple of links of those online browsers that you mentioned, or the initiatives also for grants, that could be great. And then if you send them to us, then we can share it with everyone at the end. So sure, thanks a million.
Sure.
So the second part, sorry?
No, I'm sorry. Just thank you again, all the organizer and the people to have, the patience to hear a guy from another field. So I hope that is both, clear enough and I would not say inspiring, because it will be too much, but let's say useful. Let's say something. Cross-fertilization is really the key, I think. So I hope that it will be useful. Let's say. And obviously, thanks again.
And I appreciate your effort in not complicating things, because as I had instructed Giacomo, as soon as you say words like molecule, I would already get lost. So thank you for keeping things on the data side. Even though I would love to hear more about the bioinformatics side and the theory there. Now the next part, Aaron is going to manage that. And the title is Current Challenges with TRF Analysis. And if I understand correctly, we will try to transition into Q&A straight away, since they are related.
Yeah, so we were thinking about just having a couple of discussion points here about the current challenges, and we can certainly go through that. I thought it would be good to combine this with the Q&A session, since it's basically the same thing. And for the interest of time, I thought we might start with Q&A if people have any pressing questions. I saw a jackpot of questions from Vishal in the CNSP channel, and if you want to ask one of those, great. if you want to wait, I have some discussion points that we can use as icebreakers, but someone raise their hand and chat if you want. All right, so maybe I'll get started with an icebreaker here. This is just the first of 4 points that we have, and we don't have to stick to these points. So how do we deal with small or noisy data sets that don't produce good prediction correlations? Like when you aren't able to tune, can we still use them? How do we approach that? So one way that we've been doing this is by doing generic models. So particularly on like on cohorts that are a bit challenging to work with, that may only sit still for a few minutes, like children. The idea is to get a small amount of data from a large number of individuals and then average their models. So you create probably bad models and then average them together to get a good model. So what do people think about that? Another example is to just use a standard lambda across all participants. What do people think about that? So I'm just going to leave it open for a discussion there.
I was actually wondering about the last point that you made now, about the lambda, basically, because I remember that we've been talking about it 2 days ago on the first day. Whether you have any ideas on what actually influences the lambda? Is it the data quality of the data? Why do different participants have different lambdas? Are there any ideas why it's different?
Yeah, so to me, it seems like, so I think of lambda as a smoothing feature or a smoothing parameter. So if you have a particularly noisy data set, it might require a bit more lambda. Also, if your stimulus input features are different, they might require different lambda. Like if you have a continuous model or a stick model, like the envelope or the spectrogram versus phoneme onsets or phonetic feature onsets, those might require different lambda. Also, if your stimulus feature if they're in different frequency spaces. So if you have, say, or if your EEG is in different frequency spaces for that matter, like if you input regular EEG to get regressed, or if you're inputting the power of a specific EEG band like alpha, the alpha power can often be lower frequency or is lower frequency than your regular low frequency EEG. So that might require a different lambda. What do others think?
Ola made an interesting point about the amount of data. So that's I would say an effect of what regularization should do. So if you don't have enough data to address the number of parameters in your model, that usually requires more regularization and results in a like sharper tuning curve, but also a larger lambda value. So yeah, I mean, that's a really good point. I guess another thought that I had honestly just now is some variability actually could come from the EEG spectrum. Like I noticed that subjects have more power in alpha or delta bands. And if you have different magnitudes at different frequencies, it affects the weighting of different frequencies, and that would also affect lambda sort of irrespective of the noisiness. I guess there are a lot of reasons.
Related to that point, I hope people out there know of this YouTube channel called StatQuest, mostly because of the great songs that they have at the beginning of each video, and the dry sense of humor that the guy has. But I'm putting a video in the chat that talks about Rich ridge regression and how the regularization parameter affects ridge regression. It doesn't quite, it's not quite specific to our TRF analysis, but it gives a great example of how adding a regularization parameter can prevent overfitting when you're doing, say, like no lag regression. So that's kind of what ridge regression is that he's talking about, is when the two regressors have no lag relative to each other. And you can think of EEG as being noisy and having a lot of values that shouldn't have any bearing on your regression like solution. And this video is just really great at explaining how the Lambda will solve that issue for you. And he actually has in that video a really cool example of how you can solve regression with only one data point, which is really cool. Just reading Brandon Paul's comments here. Didn't realize you're out there, Brandon. Good to have you.
I think
that's
Heather has
cool.
a question. I don't know if you wanted to ask that.
Yeah, it's related to the idea of averaging across individuals versus calculating the model at single cell. And so I'm thinking, with children or with groups where you anticipate some sort of similar response across individuals, then an average model makes sense. But like I'm working with people who have either had a stroke or who have neurodegenerative disease. And so across individuals, we expect different responses to different acoustic and linguistic features. And so there, I worry that, the individual differences are the feature of interest. And so I'm having trouble, like, if we get different lambda values across individuals, or if in the control group, I'm getting one lambda is preferable to the lambdas that are being selected across individuals in the patient group, how these different factors impact my outcomes. And also I can average across the control participants, but maybe it makes less sense to do so in the patient group. So just kind of thinking of all of the things you're talking about now and how to identify the optimal parameters, how do I identify the optimal model, et cetera.
Yeah, that's a tough one. Are you expecting their pathology to have a bearing on their acoustic and linguistic processing? So those tumors are like going to be in auditory areas then?
Sorry, what was that?
The tumors or whatever is going to be in like auditory areas?
Or so they could have they could have atrophy in auditory areas, in frontal areas, and more posterior areas. So it varies across individuals, and it also varies depending on, like for a stroke, the size of the stroke, the location of the stroke. For neurodegenerative disease, how far along in the disease are they? Where is the like focal atrophy that is then spreading out? Like all of these different kinds of things.
That's an interesting question. I don't know if I have an answer, but I do think that a generic model is probably not your way to go if you're interested in subject specific, though you might, I don't know, maybe the deviations might be informative from a generic model. Like if you try to fit someone and they are well predicted by a generic model, that's saying something about their neural processing. It's saying that they are very similar to other people. So that might be informative there, but in terms of like understanding their like individual differences, gotta have a subject specific model. And so it's not surprising that with some sort of pathology, you're getting different lambda. Don't know if does anyone else have any thoughts on that?
I kind of did. It's a really interesting problem. But it made me think of something, another way you could get generic models which is to use, and another reason for open data is to use like a separate data set that is also baby recording speech with a similar EEG setup, and you compute a generic model on the, I guess, normal hearing listeners or something that's with a control, and then you can apply it to your data. So you'd expect that people who are closer to the control cohort would exhibit like better accuracies using that model. And you might find interesting, like Aaron was saying, you might find interesting differences by trying to apply that model to this clinical cohort and seeing what those differences are. And so it also lets you separate out the modeling stage from within the data set you're working with to come up with some model that's built on something aside from the data you're working with. So then you can better compare controls and this cohort you're working with.
I like this suggestion about how, can we pose this question as how we disentangle influences of pathology versus data quality or SNR. Usually when we're talking about generic models, we're talking about data quality or lack of data, or, you know, I don't really know how to pose this question from a pathology. That is a good point, Vinay. Joe, do you have something?
Yeah, I mean, it's not that TRF has to be the solution to everything, right? I mean, there is one element which is genetic modeling. It's an approach to measure consistency. Consistency between, for example, if you have a pathology and they have a typical group, for example, you can have your model of the pathology and you can use that for saying, okay, is this group more similar to the pathology or more group or more similar to the typical group? And you can use that as a sort of a marker. So genetic models are a way to get more data, to get some subject-specific numbers that, however, tell you something about consistency rather than something that is inherently subject-specific. But there is another aspect. So think about it. I see the question from Vinay there. I just think about another situation on rather than the lambda, we talk about the time lag, for example. And so take, two populations, two groups, and you're like, oh, okay, I imagine that maybe this group takes longer to process this particular type of stimulus than the other group. And so there your output would be, instead of the time lag window being just your input, let's say, and you're going to be like, how do I compare the two groups that have a different optimal lag window? So in that case, the lag window is your output, is your result. And so you can have a sort of search, a search routine, a search iteration that finds the best configuration, the best lag window for that particular subject, the particular group. And of course, there is the same problem of SNR, because if you have a longer window, it's harder to fit the model because you have more parameters. And so it's the same question there, Vinay. Instead of having lambda, you replace time lag window, but your output is the lag window. So the lambda could be the output as well. My issue with that is that it's easier to think about the time lag window as an output because I have specific hypotheses on that. While on the lag, it's a little bit more complicated to interpret what we are getting because lambda is mostly dominated by noise. But as I said yesterday or 2 days ago, it would be cool to have lambda as an output. I just think you could have other ways to go. There could be MCCA, for example, as one approach to study data sets like that. That is completely non-TRF. But yeah, I don't have a direct answer.
I did have one thought, maybe. Go ahead.
I was just so with that, how would that compare to so looking at time lag window as an output versus standard window across all individuals, and then just comparing the time course itself? What would be the benefit of doing this windowed approach or the lag output approach?
Well, it's just I'm just making an example on the hypothesis. Imagine that you have babies, and you assume that as they develop, they start processing progressively more longer auditory structures, and so maybe they start showing some components, some evoked component that are at longer latency. And so if you take a window between 0 and 200, maybe it's better than a longer window because there is just noise afterwards. But when they develop and they are older and they have a more sophisticated neural network, then you'll see the benefit of that longer window. So in that case, you can maybe correlate the length of the time window with the development. I'm not talking about a particular paper or scientific result. It's just I'm just making it up. But in that case, the window would be your output and your hypothesis. You could do the same on the lambda. I just find it a bit more complicated to think about what exactly is the hypothesis there. But I could imagine that there could be something one could demonstrate.
And conversely, you might expect someone who has like a neurodegenerative disease to have longer time lags that where they show the most correlation. So maybe like single lag analysis might get you somewhat there. But I think important is that just like fitting generic models and using a generic model to predict someone's brain activity sort of gets at like how consistent things are across people. The TRF itself, when you do a cross validation, it's sort of testing how consistent the brain response is across that person. So one suggestion is to not skimp on data if at all possible, because when you fit single subject models, you're going to want to do a lot of within subject testing. Well, I had a second point that I just completely lost. Cool. Yeah, so I mean, just use the within subject TRF as like to test it against its that subject's consistency. So you expect the subject to be really inconsistent. You'll see that across different across the different folds in the TRF cross validation. So I mean, that might be a way that you can look at like within subject consistency is compare like across different trials, their ability to be predicted by other trials. Judith?
Going back to the window latency and so on, I was wondering specifically if you have a group that is clinically of interest or like has some pathology. Usually pathological samples, they have a lot more noise in their data as well. So how would you distinguish that it's actually a lag or window effect versus just simply there is more inherent noise and therefore there is a worse entrainment or like, there are other features happening?
I think I would go back to the within subject consistency. If the subject can consistently predict their own data, then I think that you have a good evidence that you are getting the appropriate brain response. And then you can look at the TRF comparing a non-pathological group and a pathological group. Maybe the pathological group has components that occur later. But I think it comes down to like how well can you predict like unseen brain activity with it, like within a person. Anyone else have anything to add to that? I feel like there's a lot that could be added to that I don't know. I'm sorry? Was that a normal volume for everyone else? Is that just my problem?
I cannot hear Judith, but I could hear you, Aaron.
I can barely hear you, Judith. It's really quiet. while we're waiting, Vinay had a good point that the latency is a good way of distinguishing between groups as opposed to like the magnitude of TRF, because the magnitude is definitely tied to the regularization parameter. And in fact, that's exactly what the regularization parameter is doing, is decreasing the TRF amplitude to prevent overfitting. Judith, did you get sorted?
I think so. Can you hear me now?
Yeah.
Yeah. So, well, I lost my track now. Where was I? Sorry, just gosh. Okay. Sorry, I completely blanked out now. Sorry.
It's okay. We can come back if you think about it later.
Yeah.
I actually had another thought, just a pretty general one about the pathology versus data quality problem, which is a really interesting one that I'm still not sure has a good answer. But I did think of, like, if you, aside from just using TRFs, I know a lot of studies are pretty aggressive sometimes in rejecting data and seeing how much it changes results. I remember seeing Rebecca Sachs has something when she was recording from MRI in babies. So, and I think that's one option. Like, if there is a way you can quantify noisiness in the data, lots of motor movements or something like that, and rejecting trials that you expect are particularly noisy, if you still see the same types of results, that suggests that it's not necessarily the noisiness. But yeah, it's a tough problem. I will keep thinking about it.
So I'm wondering if you would ever expect for pathology to cause brain activity to be very inconsistent to some stimulus. Like, is a TRF always going to look the same in a person? Like, if you have asked someone with some pathology to listen to an audiobook and you TRF the envelope of the audiobook with their brain activity, like, is there any time that you might expect that TRF to look different? Like the ground truth TRF to be extremely variable?
Yeah, that's a great question. That's one of the things we're looking at right now, is if we have somebody come back 2 weeks from their first session, do we have good reliability across sessions for envelope tracking or something like this? So
I mean,
it's an empirical question.
I was even thinking, like, before we get to the empirical part of that, like, is there an expectation that the brain activity could be like drastically different just when you're listening to like beeps and boops? Like, is there any impediment to like fitting a TRF that's like outside of the noise that their brains might produce? Or is the noise like part of the brain response? Does that make sense?
Can you define noise?
Yeah, okay, maybe the noise is part of the signal there. I guess that's kind of my question, is like, if you have a participant that produces a lot of noisy data, is that noisy data interesting, or is that noise interesting? Is the variance interesting, or is that just, I mean, is that just like an epiphenomenon from some injury or from some pathology? Like, is the real ground truth brain process intact and it's just noise on top of that, or do we need to measure that noise?
You know, when you talk about, Heather there in the chat was mentioning also test, retest. You know, everything you do has assumptions. So can you really assume when you have beeps and boops, maybe it's fine, but when you have speech, can I really assume that the response is going to be the same? No, you can't. Like you can do it, but it's wrong, of course. And the response is not going to be the same. And maybe that difference is interesting. You can ignore it, as we often do. But maybe that's where we find also an effect. Is that effect in the neural signal, or is it just an effect of attention, for example? That's another problem. Regarding the babies, I agree with Nate. Like I'm working on a baby data set now, and I don't know if Adam is on the call, but generally you tend to exclude lots of potentially interesting stuff. ASR works really great, Nate, and it's a recent sort of thing in a way on baby data, and I saw great results there. But when on the same data, I saw people doing analysis like the more traditional analysis, they were rejecting a lot of stuff. Well, when I was running TRF analysis, then I was trying to be the most conservative as possible. And so the TRF is relatively flexible and allow you to do less kind of rejection. But at the same time, what is it that you're studying? And so you go back to what Aaron was saying, and also if you have 3 sessions where you have the same person listening to the same thing all the time, are you looking at that variability or not? I think it's a theoretical question, Aaron. Again, what is the noise? It depends on your question, what the noise is.
I want to disagree a little bit with something, or maybe like nuance it a little bit. I don't think that, so you said that like, should we expect the same response to speech every time? And you said no, but I think that we should expect the same response, or at least the TRF is kind of, that's sort of implicit in the TRF, is that you are trying to find the most consistent response, right? So you take some set of trials and you make a model for it, and then you try to predict some left out trial. And if you can predict that left out trial, success. And if you can't, not success, right? And so maybe like when you were saying that we shouldn't expect the same response all the time to speech, maybe a better way of saying that, to nuance that, is that there might be some variance that changes as we listen to speech that can be attributed to other factors. And the TRF that we fit just hasn't accounted for those factors. And so like when you do an envelope response, you're getting just like how the brain is responding to the energetic fluctuations of the envelope, and maybe some things that are correlated with those. But you can also input something into that model that might take some of that like correlated variance away from the envelope model. Say you insert some like phoneme timing information, which is correlated with the envelope, but now you're taking some of that variance away from the envelope, and then taking some of that other variance that you can't account for with that envelope model. What you've called the responses that are variable as we listen to speech, and now you're accounting for them to some degree. But then there's always going to be that E at the end, the plus E, and
Yeah, can I give a quick reply? Then I see lots of hands up. But it's all about your questions and your hypothesis. So yeah. if now you get the person to listen to The Old Man and the Sea from a single speaker, of course your TRF is going to represent that speaker. So the envelope TRF in that case is capturing the speaker. But if you have a data set with a number of speakers, for example, then the TRF is going to be more of an envelope TRF in general, right? So and then you'll have, of course, your assumption is always like an assumption of a consistency over time. What I'm saying is that the assumption is wrong. It's true in the sense that that's maybe what you're looking for, but maybe in that variability there is one of the goals of your analysis. So if you take the work from Betash, for example, or from also Daniel Wong, for example, when he was doing his cocktail party decoding, he was accounting for those changes over time, which may be due to noise, or maybe due to other factors like attention that may be noise for certain analysis, but it may be interesting for others. Because of course, attention changes over time. And so maybe that's something you want to track. I'm just saying, sometimes noise is something you don't want, but in that noise, there may be something interesting, but it depends on the specific question. Sorry if I'm not giving any answer. I'm just saying, And the TRF is assuming something that is just, that suits us. So that just make sure that we are making the right assumption. Either was first, I think.
Yeah, I was just thinking again about consistency. And so there have been a few papers recently on the clinical relevance of TRF outcomes, thinking about whether TRF outcomes relate to, I don't know, the ability to comprehend speech and noise, or whether it can be used in clinical characterization. And so in that sense, there must be some consistency, otherwise it's not going to be a particularly useful outcome measure. And so we want to, do our best to control for factors like attentional influences on outcomes. And so if we have patients that are going to be, we need as much data as possible, let's say 30 minutes, we need to give them breaks along the way. We need to do things on our end to try to prevent some of those lapses in attention. But at the same time, there's only so much you can do, depending on the severity of the language or acoustic or speech processing deficit or whatever. We'll see differences even within a session across time. The stimuli we choose, so like we tried using Alice's Adventures in Wonderland, that's not a really useful story for the patients. They had a really hard time with it, especially given the nonsensical nature of the story. So we moved to something like nonfiction that's written for a 6th grade audience that had been validated in stroke-induced aphasia by Stephen Wilson. So thinking about all of the things we can do on the front end to minimize that kind of variability, that's a really good thing to do. But we're still just having a really hard time fitting, like our acoustic models look fine. Our linguistic models are just not getting any better than chance no matter what we're doing. And so I'm just really struggling to figure out all of the ways that working, like, is this really going to be a clinically useful approach? And right now I'm just not sure. So that was a lot and sort of related to what we were talking about.
I suppose, there are a few technical questions there, like if you say the linguistic metrics are not working, is it a theoretical issue there, or is it more of a practical issue, like do you have enough data, or, and also the nature of the data, are the participants, is the data super noisy? Why is that result not coming up? Could be a technical thing there.
Yeah, that's what we're trying to figure out. We have we have 30 minutes of data for each person. We've tried fitting various ways. We've tried changing the time windows, the regularization parameters, looking at individuals, looking at the group, looking at older adults who don't have aphasia or other language disorders. So yeah, we're trying to figure out, like, technically, are we doing things the way that we should be doing them? But it's also not clear to me whether, if we don't know about how reliable the measures are, and we don't know whether, like, there's no assessment of the validity of the measures, yeah, lots of open questions, I guess.
What linguistic measures are you using?
Oh, sorry. I don't mean to hog the conversation. So we've tried phonetic features, phonemes, lexical frequency, cohort entropy, surprisal, semantic dissimilarity.
So the kitchen sink.
Yeah, we've tried lots of things, lots of time windows, lots of changes in the regularization, print, the lambdas, all, yeah.
Have you produced a successful TRF on a not?
The envelope.
But
Envelope looks good.
On control participants.
On controls, we're not getting, so I started collecting undergrads to try to see, will the linguistic models pan out in a cleaner data set? And so far, not so much.
Maybe it's the timing of your, like, models. Maybe I don't know much about doing this, but when you transcribe a model, like the timing isn't always perfect, and you have to go in and manually adjust the timings.
We did.
Oh,
Yeah, so I think that there's something that I need to figure out, but the bigger question was still just like, how do these different, like there's so many parameters that you can adjust, and how does that impact the more general claims that we can make with these models and patient groups and control groups and et cetera.
yeah. So, I mean, there's not really, I mean, I don't really think about the TRF having a lot of parameters in terms of like fitting a generative model. It has, in my mind, really one parameter, and that's lambda, and that parameter is to like prevent overfitting or to maybe whatever. But the whole point of that is just to get the optimal linear regression solution. And like there's no, yeah. So if you're like looking at an appropriate range of lambdas, like I don't know, it's tough.
I think it could be useful to move on also to Bjorn there, who has a question for a while. But Heather, I think that it could be a nice question to explore in a separate call. Often, I'm working with those models with my students now, and sometimes it's a technical thing. Sometimes it's actually a result that things don't work. So, for example, in Michael Broderick's paper, Scientific Reports 2019 or something, I can't remember, the surprise of the semantic dissimilarity wasn't working on older adults. It wasn't working, and with a different model of surprise instead. So instead of dissimilarity to surprise, then it was working. And so in that case, it was a result that it wasn't working. Then it's better to have a hypothesis beforehand, of course, but it's tricky. So, but for the technical aspects, we could have a separate call with me or with one of the others, of course.
Yeah, Bjorn, and then maybe Michelle.
So I just wanted, it's not a response to this discussion now, it's a new question which I raised already in the Slack chat. And that is referred to when I have a competing speaker paradigm. Then what we normally do is, and that's what also suggests the MTF toolbox, right? So you train the model on the attended speaker, and then you predict the attended and the ignored speech envelope and look which one is better predicted. And the question one is, has there any attempt basically been on training the model on the attended and the ignored simultaneously? That's question number one. And second question, which is part of it, is, okay, if I have now the feature, basically the speech envelope of the attended speaker, and I've got the feature of the ignored speaker, and I use the MTRF and maybe also the banded ridge regression, do I get an individual TRF for both the attended and the ignored? And does this TRF somehow also take into account overlap correction, as, for example, the Unfold toolbox does? Yeah, these are the two questions that I would like to have answered.
So if you use banded ridge, or if you don't use banded ridge and you input two features, you'll get a TRF for both feature. What is overlap correction? Is that what I was talking about with like the shared variance between features, or is it something different?
So I think the idea is in general that if I have an event happening, let's say I think the classical example of the Unfold toolbox is there is something being presented, let's say, for example, tone, and then there is a blink, and both the blink and the tone have some, let's say, ERP or some response. But in the EEG, I only see the, let's say, supercomposition of both of these responses. And if I have, let's say, the design matrix in the MTRF toolbox, which has both features, does that somehow account to disentangling the response to each of it, and somehow this overlap. Did that make it a bit clearer?
Yes, yeah. I think it can go a long way of disentangling features that you input, especially when they're superimposed on each other in the EEG. But if you do this with two envelopes that the person is hearing at the same time, I think it's going to have a lot harder time than separate features from the same stimuli. So for example, the spectrogram and the phonetic features, if you fit those individually, sometimes they'll capture some of the variance from the other feature. I'm not quite sure, since some of the response should be fairly similar between an attended and an unattended speaker. I'm not sure how the TRF would actually handle that. Maybe.
Yeah, I mean, in general, the question comes from the point whether if I include both, because the participant is listening to both simultaneously, can I better predict the EEG? I mean, that's the underlying question.
I mean, you should be able to better predict the EEG anyway, because even if there is some shared, so I'm imagining that that attended speech and unattended speech have some sort of similar low-level processing going on, and then maybe attended has a little bit different high-level processing or something like that. So even if there is a bit of competition between the two features and the TRF model to capture that similar low-level processing going on, I think that by having both, you're going to capture both that shared bit and something that's unique to the attended, or even if there is something, especially if there's something unique to the unattended, I think that you're going to get a little bit more. So you're definitely going to predict better by including both features. And even beyond that, or even before that, you'll have a bit of, because you have a second feature, you'll do a little bit better. But I'm not exactly sure how that shared part or the similar low-level responses would be divided between attended and unattended features. Is a TRF expert out there like have any thoughts on that? No, I don't know.
I got distracted with the chat. What's the question?
So if you have an auditory attention decoding paradigm, subject hears two envelopes. If you do a forward TRF model using both of those envelopes as predictor features, we think it's going to do better. I presume it's going to do better because it's accounting at least for the shared part and any unique parts that are out there. But if there is a shared component to attended speech and unattended speech, how do you think one or the other might suffer? The TRF, I mean, how would the attended TRF or unattended TRF suffer? Like, is it going to produce good TRFs for both features?
I mean, you could fit a TRF with three components, with the envelope of the attended, of the unattended, and the combined envelope. Maybe in that way you'll separate more, but I'm just thinking about CCA. That's what we did in that case, right? The point was that you have all of that at once, and then maybe you'll get one component that is more attended and one component that is more unattended. And so there you're separating. With the TRF. Yeah, maybe it will suffer a bit. Yeah,
I think if you include the three feature model with what the subject heard, attended and unattended, you're going, the what the subject heard model is probably going to soak up all of the same variance. It's just going to be an acoustic model, like how did the energy affect brain activity, and then maybe your attended and unattended will have their unique parts. It's a good question though, something I don't think I've ever really thought about.
just that generally forward models are not as good for decoding anyway.
Yeah, that's true.
So that's the thing. But you know, from the theoretical perspective, it makes absolute sense.
Does that answer your question? Did you have another part of the question, Bjorn?
No, that definitely answers my question. Yeah, and so the second part for sure. Oh wait, one of it at least. So about this overlap correction, I'm still not sure because I remember having a discussion with Benedikt Ehinger, who did the Unfold toolbox, and he said that they so far only use very sparse design matrices. So they only have onsets or so they don't have continuous stimuli. And they were quite, if I remember correctly, I don't fully understand it for sure. They were saying if I've got these continuous features, then the overlap correction becomes difficult because of the regularization. But that's as far as I remember. So I don't know whether you can comment on that.
I feel like that's a bit above my pay grade, honestly. I don't know if I really quite understand the problem.
Yeah, okay. Maybe we can have it at the discussion at another point as well. Because I also don't understand it to explain it well enough. So
Fair. Vishal, you've had your hand up for a long time, and I'm sorry.
Yeah, no worries. I hope you can hear me well.
You can.
Cool. Yeah, so I just wanted to resurface the question that I asked in Slack.
Yeah, you've got a lot in there, and I like them.
Yeah, so maybe if you could probably take them on their own pace, that would be great. So.
I'm sorry. What was that last part?
I just wanted to bring them up again from the Slack. So
do you have a favorite question that you want to ask?
Yeah, I think it's all in the order of the priority. So I think the most important question that I actually wanted to ask is the way MTRFs are computed under the hood. Because if I understand correctly, the implementation is that you have models fit for each and every trial, and then you try to average the weights for all the trials in the training set, and then you try to return that as the final model, right? So how would that compare to jointly optimizing over all the training trials in one go?
So I actually do have an answer for that one, and the answer is it matters. And I also think that the latest version of the toolbox on Mix GitHub should not be averaging across trials or across models. The reason it matters is because if you have trials, or I guess especially if you're working with a generic model, if you had subjects with different amounts of variance in their EEG data, that could affect your results. If you're working with forward models, I actually think this is perhaps less of an issue because there's consistency across the structure of speech if it's one audiobook that people are listening to. But if you're listening to like multiple types of music, so one study I was doing was like different rock songs which have different spectra, that's where it matters. The key is that when the TRF is computed, you need this covariance matrix XTX that's multiplying your input. And so that when you average the models, you're assuming you can average those XTX matrices. So the best option is, which I think the newest toolbox does, is to compute a model on all of the trials together rather than averaging each of them. That's the safest way of dealing with that issue.
Sounds good. Thank you. Yeah, and I also want to hear from the community as a whole, when it comes to what are the best practices when it comes to applying filters for the EEG data before you do TRFs, right? So I just wanted to know, maybe Giovanni, if you also have some thoughts based on your discussion with Alan, you could also probably jump in on this.
Don't use filtering. Now, Alan has been really clear, I think, in his Neuron paper a couple of years ago on the risks that come with filtering. So filtering is going to impact your data instead. Every time you filter your data, is going to introduce some noise of some kind. And that depends on the impulse response and on the step response of your filter. So we tend to use filter anyway. So you filter both ways so that the delay in the signal at least is not a problem, but the signal is always going to be smoothed around the zero. Now, and because you use filter, you're filtering twice. So if you use an IIR filter, you need to use a very low order filter. If you use an order 8, then it becomes 16. And so I use filters of order 2, 4 maximum. But so Alan is not that he says don't use filtering, because of course, in certain cases, it's important to use it. He just says, be aware of the risks. So when you apply a filter, whatever filter there is no wrong filter per se. But when you run a filtering step, then you should be aware of the impulse response of your filter. And you can get that just by building a zero vector with an impulse, maybe in the middle of the vector, and you just pass it through the same filter, and you study the effect of that particular filter on an impulse. That's going to tell you about how the shape and also the width of the artifact. If the shape and width of the artifact are similar to the TRF you get afterwards, then it's a problem. Of course, if you apply a filter, what I'm saying, like a Butterworth filter order 2, and you study that, you study the impulse response that maybe has a impact of the order of 20 milliseconds. So a response that looks like a TRF, but is 20 milliseconds of impact. The TRF that we get have a duration of, and you have a response at 80, 100, 200, and so on. So the 20 millisecond of filter, it can't explain your response. So you need to be aware of the impulse response of your filter every single time you run a filter. That's also why I tend to use, maybe 2 filters in the same study. I use 2 filters with different impulse response to check the input impact of the filter. Not everyone has to be a nerd of filters, but you can also try without filtering. So there are detrending functions that are in fact practically doing an interpolation of your data, a fit of your data, and subtracting that fit to your data. So if your data has a linear drift, it fits a line of a particular, in that case of an order 1, and it just subtracts that. And so it's not filtering, it's a detrend. It's going to cause an artifact as well, a different type of artifact. But it's important to be aware of the artifact. That's my answer. Does that answer the question?
Yeah, I think that makes a lot of sense, because I think looking into the impulse response is much more informative way of looking at the effect of the filter than applying it without looking at it. So yeah, I think I'll learn to do that from now onwards. That's also a good thing. Also in the CNSP resources website, right? So I just looked into a bunch of functions that you have, like you have get HPF, get LPF. So these are all like Butterworth filters of order 2 or 4 at max, right? So is this the same implementation that you were talking about just now?
Yeah, exactly. I mean, those are the filters I started to use, and I have nothing against Hamming windows or other filters. That's just my go-to sort of first filter I use, just because I know the impulse response. But then you should not get stuck to one. Correct. Like you shouldn't do it, or always use the same thing. Otherwise, the risk is that you're just studying the response of the filter. So there is not an easy answer. There's not an answer like, oh, you should use this filter, it's always going to be fine. That doesn't exist. So you need to be aware of the impulse response of your filter. Even if you're not an engineer, it's still easy to say, okay, I take a zero vector, I put an impulse there, and I look what the result is. And that shouldn't be the response that you get in your TRF.
Right.
Can I answer quickly? There was a question there.
Sounds good.
And then I know you have other questions, Vishal. I just wanted to, before it gets lost in the chat, it was asking if you do an experiment with 3-second long trials and with 30-second long trials, is it the same from the computational perspective? And Nate said, and I agree, that's great. The TRF now doesn't do averages, doesn't fit individual TRFs and then do the average of that as it was originally. And now it's just taking all the data, is playing around with the covariance matrices. But the point is that if you have 3-second trials and your TRF window is 1 second long, let's say, I'm exaggerating, let's say it's 1 second long. Of course, to fit a model that has a window of 1 second, if your trial is 3 seconds, you're losing that 1 second. Because if you're, you need to take, when you take the lags, you either pad your envelope or you chop your EEG or vice versa. And, because if the envelope in one point causes the response on the other second, you have to stop a second before, basically. So if you take very short trials, you're going to lose the window at every trial. So at the end of the day, you're going to lose more data if you have shorter trials. So computationally is going to matter if you have very short trials. And the other aspect is, neurophysiologically speaking, you're going to get a massive acoustic onset response if you have short trials. And I tried with very short trials, also with the cocktail party thing. Cocktail party, you have 2 different voices talking, and you have a very short trial of 2 seconds or something. And it's very hard to see much because there is this massive onset response everywhere. So there is also that aspect. But this is just my view. I shut up now because I talk too much.
Judith, do you have a question?
Yeah, actually following up on the trial length, I was wondering, because you mentioned that you're now working with some baby data as well, and we're setting up some experiments, and we were thinking of using 20-second trials. But the thing with baby data is that it's unlikely that they are going to sit still for 20 seconds, say 15 minutes. So then you end up like chunking like different things and then concatenating it. And I don't know to what extent actually that's a good idea for, I don't know, what's your recommendation?
Yeah, I mean, what we did was I mean, I didn't collect the data. That's not my output. I was just analyzing there. They collected also eye tracking, if I remember correctly there. And so basically they knew when the baby was looking or not looking at the screen. So in my case, like, we didn't really care. We just fit, it was auditory. The part we were analyzing was auditory, so we were just fitting everything. So what do you mean by cutting the data? And like, were you just removing the parts where we were not attending, they were not looking? But was it auditory?
No, this is just, we are setting up the experiment, but I have a colleague who has done some work before and essentially say that you look at your EEG data, and like if you have too much artifacts or whatnot, you chunk your good quality data. So then you're only using the stimuli from, so although you have 15 minutes of baby, actually you might have, I don't know, 10 minutes of good quality data. So it's not continuous per se.
Okay, sorry, now I got it. Yeah, no, in our case, we were using ASR denoising, which we found super beneficial for that.
So you keep all the data no matter what.
And we kept all the data, yes. So in that case, it was an auditory experiment. And so even if the data was super noisy, they were still hearing the thing. And so we just assumed that it was going to be okay. You could try to regress it out if you had some accelerometer or some EMG or something. You could try to use, for example, CCA to regress it out. But you could also chunking it, you could also remove it. That's true.
I would be careful about.
Trials, sorry.
Go ahead.
I was just going to say that if it's a good idea to keep the trials at 20 seconds, or if there are 6 trials that belong to one condition, is it better to concatenate all the data and the trials?
Well, I wouldn't concatenate them. Like I would put them in the same, I would feed them through the TRF as the same structure. I wouldn't put them, concatenate them physically, you know what I mean? But if there is a way to capture the condition as a regressor, then it could be great to have one model, because you can capture everything, and this extra regressor is going to capture this distinction. What are the conditions?
It's auditory and audiovisual, basically, and then native speech and foreign speech.
That's for MEC then.
Joe, did you say that you would put a feature in that would identify the discontinuity? I think I just missed what you said.
No, for the conditions, if she has two conditions, maybe you can have a regressor that captures the distinction between the two conditions, so you can fit a single TRF. But I mean, if you don't have enough data for each individual condition, like if you have enough data for each individual condition, I would fit the two things separately, honestly. But with baby data, I don't know if you have enough data. Nick?
Yeah.
Yeah. What was the question?
If you have 20 seconds, so 6 times 2, 12, so 2 minutes of data, is that enough to fit a model, or will you concatenate different conditions?
Sorry, could you repeat how much data you have? I'm finding it very hard to hear you.
Yeah, sorry. So say 20 seconds is for each trial, and there are 6 trials in total in one condition.
And you're saying, should you combine the data across conditions?
Yeah, so if we repeat this same condition later on, is it better to just get like all the trials together?
Like in terms of fitting the model, or just in terms of tuning the parameters of the model?
In terms of fitting the model itself. So I guess the question was kind of answering that it's just better to keep the trials separate and not concatenate them, right? Because you want to maintain things continuous.
Well, yeah, I think you have to do it separately. Because I mean, what are you trying to do? Like, what are you trying to demonstrate with them?
Well, no, it's just that I was thinking that 20 seconds wouldn't be enough for a trial, and we can't have a baby like listening to something for longer than that. So I was thinking that if you actually concatenate the trials, it will be more useful. Within
condition.
Yeah.
Yeah. Yeah, I mean, you can concatenate them. You don't have to physically concatenate them. You can, I mean, depending on what you're using, what software you're using, but you can have sum your covariance matrices across trials, which is a better way to do it. That way, there's no kind of discontinuities between the EEG data.
Those discontinuities will produce TRF artifacts, by the way. So when and you can see sort of the effect of that. Well, that's more of a filtering thing, but I was going to say that you could see more of an effect of that in Alain's paper. But I think it's fairly similar, because you've got just a big high frequency something, and the regression algorithm wants to, especially like at non-zero lags, it's going to want to fit some component to that discontinuity. So putting them in the same, so the input to the TRF is a cell. Putting multiple trials into one cell where they're like actually back to back is not a good idea. But because the TRF now sums the covariance matrices, it's 20 seconds is not too short for a trial, I don't think. I've had pretty good success doing 30-second trials. And I know people in the lab that have tried with 8 and 10-second trials, mostly just fine.
Yeah, the length of the trial is irrelevant. It's the number of trials and the cumulative amount of data, you know. So
like lots and lots of very short trials, not very short trials. I mean, there's probably a lower bound on this, but if you can get 20-second trials and you can get a number of those trials, should be good.
In fact, having really short trials is a good thing, because I think there is a function in the M2F toolbox to actually split up your trials into shorter trials and then sum over them just to be more computationally efficient. So yeah, I mean, as long as it's large enough to deal with the type of window you're using, integrated window, then that's fine. Yeah, it doesn't matter. But you know, you want to make sure that you're getting more than maybe 15 or 20 minutes of data in total per condition, if possible.
And in the absence of that, maybe like you could explore generic models. That's something that we've done when you can't get that much data. Like if you're, if you can get about 5 minutes of data, then a generic model is pretty, that's a pretty good candidate for a generic model, as long as you're not trying to explore individual differences, because generic models obviously can't do that. And actually, so we're working on a small project right now that where we're seeing how, like, when a generic model wins, and I'm not going to give away too much of a punchline. Actually, I'm going to give away the whole punchline. It's like between 2 and 5 minutes really is when generic models win over a subject-specific model. Again, depending on how many subjects you have, because the number of subjects will influence how the quality of your generic model. So if you've got 4 to 5 minutes of data from 20 subjects, that's like right in the sweet spot.
It's all dependent on the system as well there, Aaron. Like if you have a system with low SNR, like it's not necessarily the duration of the experiment, it's the information rate. It's the amount of information you have, which lowers if you have noisier data. But so with baby data, if you have the same amount of data, it's not necessarily as the numbers are not exactly the same. But yeah, no, that sounds great. Sorry, does that answer the question, Judith? And just because there was a question in the chat from Fatime, who's in the library, so she can't speak, so I speak for her. And she was asking about the project. She says, do projects may include very new ideas, and is it possible just working on EEG signals without audio signals? So she's asking about the call for projects there. And the answer is yes and yes. So you can include very new ideas, and you can include things that are not about auditory processing, as long as it's within what we're talking about. So natural sensory processing could be lots of different stuff. So as long as it's relevant, it's absolutely fine. Sorry for the interruption, Aaron.
It's all good. If no one else has a question, I might ask this one question from Vishal. It's something that I've always found interesting, and it brings up a good point that I didn't really think of. In the 2016 paper, it was suggested to have some safe extension at the ends of the TRF window to account for edge artifacts. I believe that in most papers, these edge artifacts are chopped and not reported. Was wondering, A, what causes these artifacts in the first place, mathematically? B, when people try to predict from the learned MTRF, do they predict with the edge artifacts intact, or do they chop them off before predicting? It's an interesting phenomenon. And I have no idea what causes it. Maybe some of the other TRF experts out there can answer that. But what he's talking about is, if you haven't seen it, you predict an MTRF and you go out, say, to like -200, where you expect absolutely no activity. From 0 to 199, the values are floating along at, say, 0, where you would expect them. But at the very last, or the very earliest, I guess, sample, you either get something where all of the values are super high, all of the values are super low, or there's a big spread of the values. I have no idea what causes that. And it was my understanding that it probably wouldn't affect the prediction, but I was thinking from the perspective of train up an MTRF with long, like, pre-stimulus and post-TRF lags, so that you can just chop it off for visualization. But the question is, like, does that occur in every TRF, and does that affect the predictions? So does anyone know like what causes that like artifact at the end, the last and first sample of the TRF? So no.
So I don't have like a very good specific reason for that one, but I remember trying to figure it out for a while when I was first working with TRFs. And what I've centering, like making sure the average of the inputs and the average of the outputs is 0. So like if you're doing a forward model, making sure that the envelope has a 0 mean, or making sure that the EEG channels have a 0 mean, that does tend to reduce those edge things. Not entirely, some subjects I'll still see it, but that, from what I remember, is a big part of it. I speculate it's because it's trying to account for some offset, which just having the weights themselves doesn't necessarily deal with, because you're trying to add all of those together. That's not a really good reason, but from when I was like trying to figure it out, I found that normalizing your data usually tends to reduce them a decent amount. And when I report TRFs, I try not to, I don't truncate them. So yeah, anyway, it's a really interesting question.
I thought the TRF had a bias parameter in it, or not a bias, but the equivalent of y-intercept.
I believe it does now. I had been working with some TRF related stuff that did not have the bias parameter, but I don't know if the bias parameter captures it entirely, and I don't really have a good reason for why it wouldn't, but this is more empirical. Like I was trying different things and I remember that being a part of it. Yeah.
Can I complement? I completely agree, Nate, from what I played around with the TRFs, but my intuition there is that the assumption is that the data should be infinite. So the trial should be infinite. The shorter, the trial is not infinite, does not have infinite duration, and there's the start and the end are discontinuities in a continuous signal. So the normalization helps, as Nate said, but I think that when you fit the TRF at the end, it's as if there was a 0 padding afterwards. So you're going to have a jump. So I think that there are some discontinuities that cause that, and the normalization helps. It may not be the full answer though. So
I guess another thought that I had that I just remembered is aside from the biasing parameter and the offset, if there's like a mismatch in spectral content, like if your input, their feature has a lot of low frequency activity, but then the output doesn't, that can also produce that. I remember seeing that when I was working with trying to reconstruct envelopes and forward modeling of envelopes. So actually the spectral content of both and mismatches between those can also, I think, produce those sort of edge effect artifacts. And the other thing that I thought of is it could be something really interesting. So I actually do, if you do see that, I recommend trying to look at a larger window to see if it actually is something real. Sometimes it's not, but I did find in one paper when I was analyzing speech tracking that there is like a low frequency tracking at pretty long delays. And so it might be something interesting. So I do recommend checking that if you're interested.
Thank you. That helps a lot. But I just want to like also get what people do in the community for the point B when they predict from the learned TRFs, right? So do they chop and predict, or do they just use the actual learned TRF with the artifacts and then predict the unseen data? Is that so? What's the common best practice that people do?
So the way the TRF toolbox works is whatever inputs you put into it will be predicting used to predict your EEG data. I don't know if others go outside of that practice, because it is possible to extract the model and remove some weights from the model and then input that into the MTRF predict. And I know that, I mean, maybe not within within one model, that's not something that's common, but it is common to fit a joint model and then remove one of the TRFs from it to just get the to just use one of the models to predict the EEG to see the effect of that feature, but also to account for the other feature. But I don't know about whether other people like take away some of those what looks like splattery weights. I certainly haven't done it. And you know, this is anecdotal and subject to my bad memory, but I don't seem to notice this, what I just called splatter. I don't know why I called it splatter, but I don't seem to notice those as much during lags where like there should be brain activity going on. It's always in like really, really early or really late lags. Could be because I'm inputting like Tmin and Tmax to be early and late, but it seems like when I've looked at a model that starts at time 0, there's not as much of that explosion of the weights at time 0. So I'm not sure if it's too much of an issue, because typically when I try to predict EEG with a TRF, I don't input negative lags unless I'm expecting something occurring before to predict the EEG, which that's possible, but not very common. So I tend to exclude those really long and really early or pre-0 lags. And I think that more or less, for me at least, accounts for or removes that splattery TRF weights. Does anybody do anything else? Anything different? Cool. So some of us have to get going places, and I'm sure like you all want to have your afternoon, so or night or morning, wherever you may be. So I was going to open it up for one last round of questions. If anyone wants to ask anything else for the group, we should do that now. I don't mean to pressure anyone to not ask questions by saying we should get going. So like, please ask questions while we're here. Judith?
Sorry, this is just a hypothesis out there, crazy idea, but is there any possibility that TRFs could be used somehow to have more spatial information, in that at the moment we use every EEG channel and we get a model of like how the brain is tracking this kind of thing? But would it be possible to adapt it in such a way that you can have more information of which signal, or you have to do the analysis separately for like your regions of interest?
So EEG isn't a great method to get spatial anything. You could use the inverse problem, could find brain activity in source space and then do TRFs on source space. That is done typically in MEG and not EEG, but I mean, that is one solution I think would work. I'm not sure if there's, I mean, I'm sure there's a way of implementing some spatial constraint on TRFs. I'm not sure how that would work. or if we would even implement that. I think probably the best solution would be to do source-based TRFs.
But would you say, is it a good idea or this is a bad idea to take like channels of interest and say like split your signal, or like would you wouldn't recommend that?
I'm sorry, can you explain that more? I don't think I
So
understood.
say you have like 6 channels that are occipital, 6 that are temporal, 6 that are frontal. So
Yeah.
you basically just select those channels out of your EEG signal as input for the training model, rather than taking all your signal.
You mean as the output of the training model?
No, both for the training and then the testing. So you do basically everything only with the, say, 6 channels or 10 channels or whatever.
Oh, so you're trying to fit only on those channels instead of, that's possible.
Okay.
I mean, you can, the only reason to exclude channels from the code is for time. You can just regularize or optimize over the channels of interest too. So at the end, when you looked and see which Lambda performed the best, typically what we will do is average over trials and channels, all channels. And I think that in the absence of like any hypothesis, that's probably the preferred way of doing it. But you can select which channels you average over too.
Thank you.
Yep. Is there anyone else? Joe? Do you want to? Is that it? So yeah, like Joe said, thanks for all your participation, all your great questions. Thanks to all the speakers. Hope everyone got something out of it. It was great to do this. Anyone else want to say anything, organizers?
I mean, just thanking everyone that sticked around until, I mean, there are 27 people in the call and we went way over time here.
Yeah.
So thanks a
Thanks
million
for sticking around.
And look, I'm happy this was useful. I'd like this to be useful beyond the workshop. So get in touch. If you know, like Heather, if you have any questions on your specific experiment, get in touch. If you want to help, and we would be welcoming your help in terms of data sets, standardization, if you have any opinion on that. I like to do science. I don't like writing documentation on standardization and everything. I actually don't like it at all, but I think it's useful. So if someone else wants to be involved, that would be great. So thanks a million for participating, and the videos will be online.
I have a really quick comment. So, Joe, you have a forum set up. I wanted to make sure people know about this.
Yeah, I was gonna, yes.
I want people to know about this. So for the future, that is the place where you should post questions. He has a very nice forum set up to like catalog. Thank you. Yeah, so Giovanni just sent the link to catalog people's questions and back and forth for feedback. So, and it will stay there. So if anybody else has questions, then they can see it as well. So I recommend using that post CNSP.
Yep. So thanks again, everyone. We'll see you next time. It was a great meeting, and bon voyage.
Yeah, thanks for